Today we are going to learn mechanisms of antibiotic resistance. So in this we will see overview of the mechanism of antibiotic resistance. What is bacterial cell? So this image illustrates the four main mechanisms of antibiotic resistance, which is the ability of bacteria to resist the effect of antibiotics. These mechanisms enable bacteria to survive and multiply in the presence of antibiotics that would normally kill or inhibit their growth, posing a significant threat to public health. Bacterial cell. The central focus of the diagram is bacterial cell, depicted as a rod-shaped organism. This cell contains its genetic material, including a chromosome and plasmids within the cytoplasm, all enclosed by a cell wall. These components play crucial roles in the development and spread of antibiotic resistance. Alteration of drug target. One mechanism is the alteration of the drug target. Antibiotics work by binding to a specific target within the bacteria, such as proteins or enzymes. Bacteria can alter these targets through genetic mutations, preventing the antibiotic from binding and rendering its effectiveness. Then modified drug target. Illustrated on the left, the modified drug target is represented by a receptor that has changed its shape. This change prevents the antibiotic molecules, shown as small blue spheres, from docking with the target, thus negating the drug's effect. This is a common resistance strategy against drugs like penicillin. Then activation of drug efflux pump. Another mechanism is the activation of drug efflux pumps. These are protein channels in the bacterial cell membrane that actively transport antibiotics out of the cell before they can reach their target. This effectively lowers the intracellular concentration of the drug to sub-inhibitory levels. Then, inactivation of drug by enzymes. Bacteria can also develop resistance by producing enzymes that inactivates the antibiotic. The drug inactivating enzyme chemically modified or destroy the antibiotic molecule, preventing it from functioning. A well-known example is beta-lactamase, is the enzyme which breaks down the penicillin and other beta-lactam antibiotics. Inhibition of drug uptake. The fourth mechanism shown is the inhibition of drug uptake. Bacteria can modify their cell wall or membrane proteins known as porins to prevent or reduce the entry of antibiotics in the cell. This is particularly effective against larger antibiotic molecules. Then modified cell wall protein. This is visualized by a modified cell wall protein at the bottom of the bacterial cell. This alteration is the protein structure effectively blocks the antibiotic's entry point, reducing its accumulation inside the cell and contributing to resistance. So that's all about mechanisms of antibiotic resistance. Thank you.